are in listen-only mode.
Welcome, everyone. We're going to hold on just for a few seconds to let a few more people log in, and we will begin. Well, welcome. I am Joanne Geer. I am the Executive Director of the Biopharma Research Council, and welcome to our webinar in our series called D3D, Data, Drugs, Diagnostics, on Next Generation Biobanking. The Biopharma Research Council is an association for scientists across all the many silos of biomedical research. So we include industry, academia, nonprofit, government, and supplier labs, and those who love them. This is a sampling of some of the activities that we've done in the past year. We had a great CRISPR program in Boston in the spring, cybersecurity program in Princeton this summer. In September, we went to Frederick, Maryland, to the National Cancer Institute for a program on the microbiome. And we just got back from Durham, North Carolina for a program on technology convergence. As you can see, D3D is a theme that we've been developing for some time. We have meetups we're based in New Jersey. We have physical meetups around here and a number of webinars. And it's really about cultivating that cohort of individuals who are really working at the communication between the scientist and the IT professional, and especially those folks who have a foot on both sides. Next year we'll be doing some really interesting webinars and various themes, and I just encourage you, if you have something you're working on that you'd like to share with our audience of 9,500 scientists, please feel free to be in contact and we'll take a look and see what we can do together. And this is our holiday. It's the beginning of the holiday season, so I just wanted to share that picture with you. Today we're going to hear about a next generation biobanking in a targeted outsourcing model from Jan Wang of Biofortis and Amelia Warner from Global Specimen Solutions. I'm going to introduce them to you now so we don't have to interrupt between the talks, and then we'll take the Q&A when they're finished. Dr. Wang received his PhD in bioengineering from the University of Washington in 1996. After a brief postdoc at the Carnegie Mellon, he joined the biotechnology industry, first with Solomics, which is now part of Thermo Fisher Scientific, and then Physiome Sciences and Paradigm Genetics. He joined Biofortis in 2004 and became its president and CEO in 2005. He developed several commercial life science infrastructures with customers in academia, government, and the biopharmaceutical industry. Dr. Warner is the founder and CEO of Global Specimen Solutions, an innovative specimen management CRO in Raleigh, North Carolina. She received her doctorate from the University of North Carolina and completed residency and fellowship training at St. Jude Children's Research Hospital. She has over 15 years leadership experience in the pharmaceutical industry and academia, including head of clinical pharmacogenomics, clinical specimen management for Merck, and head of pharmacogenomics for the Children's Hospital of Philadelphia. You can enter questions at any time. We're going to address them all after the two talks so that we can really have an interactive conversation. You have a question box on your dashboard on the right-hand side of your screen. And yes, we will be recording this session, and it will be posted along with the slides within a day or two, and we'll send out an email to let you know when that's available, and you can share it with anyone you like. And we are ready now to go to Dr. Wang.
Okay, can you hear me?
Yes, we can.
Okay, good morning. So today's presentation will be a tag team between me, Jan Wang, and my friend and colleague, Amelia. So some introductory concepts, and as well as from a more informatics perspective into next generation biobanking, and Amelia will take over and go into a lot more detail on the regulatory aspects, outsourcing aspects, and so on. So again, welcome. Hopefully we'll have a very lively discussion. There's a lot to talk about. I'm going to go through them fairly quickly at a fairly high level, but feel free to dive into any level of detail during the Q&A session. Now, biobanking used to be simple. Everything is contained within your four walls. The main responsibility is to track samples in and out, and that's it. However, things started to change. I gave an interview to Nature Methods almost 6 years ago now. Really, I started to talk about you should be looking at biobanking as not as an end, but as a means to allow for translational research. It's not just about operational biobanking anymore. It's about what you can do with the samples in your biobank. Right? A biobank, to me, is a liability, not an asset, until and unless you can use the samples in your biobank for amazing things. And that's what Next Generation is about. So what makes a good sample, valuable sample? It needs to be of high quality. It needs to be highly annotated. It needs to be properly consented. Amelia will go into some details on that. And ideally, if you can link it back to patients, reach back clause in your consent, that would be wonderful. Okay? Now, how do we get to high value samples? There are some challenges. The world is shifting around us, and there are some challenges, high level challenges that we need to be aware of. Right? The first one we talked about already, which is it's not just an operational exercise anymore. It needs to drive science. But then science is also done, the way science is done is changing. Right? There's a lot of externalization, a lot of collaboration. Everything does not happen within your four walls anymore. And because of precision medicine especially, the role that samples play in clinical trials, for example, is becoming more and more important, and that's attracting increased regulatory scrutiny. Now, the way I think of Next Generation Biobanking, I think of it as three different circles overlapping with each other. There is the operational component, but there's also a virtual biobanking component, and a good illustration of virtual biobanking is really in the clinical trial sample and consent tracking space. And I will go into a little bit of detail on that. But then again, it's the data management associated with both operational and scientific aspects of your biobank, of your samples, of your clinical trial consents. That's what allows you to do amazing things and derive value from your biobank. So all three things really is part of this ecosystem that I call Next Generation Biobanking. So let's dive into some of them slightly. Most of you are probably very familiar with this picture. This is a clinical trial, looking at a clinical trial from a sample-centric perspective, where you have samples going to central labs. Sometimes they don't. They directly go to testing labs. Central labs may do certain things, and they will send it out to various labs, and leftovers or future use samples may go to long-term storage, and so on and so forth. So there's an ecosystem out there that's handling samples. And then you have the sponsors that provides additional information. The clinical trial sample and consent tracking circle really means, do I have visibility into what's going on in this ecosystem? In today's world, from what I've seen, most people don't have a real good idea of what's going on, right? A lot of exchange going on is via files in Excel, and when somebody needs to get a report on where are all the samples, when am I going to be able to do a test, where am I going to have all the samples for this time point in a clinical trial, then it's a lot of Excel shuffling, and that's problematic. This is an actual instance of how information may be tracked, may be shipped around and tracked, right? So, for example, the sponsor we'll set up a trial and define sample logistics to a vendor or CRO, and that sample collection specification is sent to the site. The site will collect samples as well as consents. Patient consent, in this case, goes back to the sponsor, and then the samples go back to the central lab. Central lab periodically would send inventory information to the sponsor, and if requested, send actual samples to the sponsor. But if there's a consent deviation, the patient withdraws consent and so on, that information needs to get to the sponsor, and the sponsor then needs to send instructions for destruction and so on the sample. Okay, so that's kind of one instance, one manifestation of the last slide, which is the ecosystem. Right? There are other types of manifestations, and remember, this is also geographically on a global scale, so that it is a form of a virtual biobank. You need to orchestrate and know what's going on geographically, in a geographically distributed fashion, as well as in a vendor distributed fashion. Okay? So how do we deal with that ecosystem? We won't go into a lot of details, but this is the conceptual framework that we've found useful. So there are three components at the conceptual level. There's the plan, there's the reality, and then there's the consent. So the plan means, I'll talk about this in the context of clinical trial. The plan means, what am I supposed to get? Okay, what samples am I supposed to collect, in which arm, at what time point, and so on, for what purpose, and where are they supposed to go? From site to the central lab, from central lab to test lab 1, and test lab 1, if they have leftovers, it goes back to central lab, or go to the storage, and so on. So that's the plan. Reality means when samples actually start to come in, what actually happened? What do you actually have? And usually you don't have, not everything is matched to the plan. And did they actually go to the right places at the right time? So that's the plan. And the consent means, what can you do with the samples? You have the samples doesn't mean you can do anything you want with the samples. Okay? On top of this framework, then you can do all kinds of interesting things. For example, reconciliation. Do I have what I'm supposed to have? If I don't, what happened? Can I intervene and fix the problem sooner? Okay. Can I request samples now for a specific analysis? For future use samples, do I have the right consent so that I can conduct a specific study without violating consent permissions, and so on and so forth? Okay. I saw this recently on LinkedIn. I just had to share it. The plan may look nice and clean. Straight line with a little bit of a curve, you can get there easily. Reality is a lot messier. In fact, reality is really messy. So you need to be able to reconcile between the messiness here with your plan there. So you need to have, again, this talk is from an informatics and system perspective, you need to have a very, very flexible system to deal with the reality. If you're expecting to deal with this, you're not going to go very far. You should be expecting to deal with this. Okay? So what's a typical process like? What's involved, right? So you have to, when you plan things, you have to plan for, you need the information about patient visits, what are the consent templates, what kind of information you need to extract out from planning purposes. The biggest problem I've seen on the planning side is that planning information is not structured. Planning information is in documents. You can't compute against documents, so that you can't really reconcile easily other than having to eyeball things, because computers deal with computable information, not a piece of paper, right? So that is the biggest challenge. That applies to both sample-oriented planning and consents. They're usually on paper. No, paper in quotes, Word document is paper, effectively, for this purpose. And then you need to understand about patient enrollment. You need to understand about getting samples from patients. You need to be able to reconcile plan versus reality. When you need to use samples, you need to be able to request, and you need to be able to actually get the samples upon request. And when the time comes, either due to consent or whatever defined parameters you have on scientific reasons that you want to destroy samples, you need to have a process to destroy your samples and certify destruction so that if you're audited, you can prove you've done the right thing. Okay. Where do we get the data to populate the necessary information to fulfill that golden triangle that I was referring to? That's the framework of how we think about this space. So your study protocol and your lab manuals are usually the source of sample collection and routing plans. Again, they're not structured usually, so you have to apply structure onto that document. Informed consent documents, Amelia will talk a lot more about this, both at the study level, regional level, site level. You need to be able to extract the correct information, codified information from consent documents, because consent documents are literally documents. You can't compute on documents the way we need to anyway, right? So these two areas are usually, as of now, what I've seen is manual work to extract information from documents. And then you look at something like an IVRS system as the first source of an identifier for a subject, because everything else hands off of an identifier of a subject. Okay? And then the EDC systems then will tell you about patient visits, the fact that this patient came in for this visit. If you compare that information with the sample collection plan, then you can know the expected what should have been collected for this patient at this visit. Okay? And EDC will also tell you the patient signed this consent at this time. Okay? And then if you have a central lab is probably the first place that you will have information about samples. That's probably where the identity of samples are first established. Yes, samples are actually created at the site, but it's pretty difficult from my experience anyway to push new systems into the sites, and dealing with hundreds of sites is not an easy thing. And EDC is not the right platform to deal with samples. There have been several attempts at doing that, and it's not the best approach. So chances are you're getting your sample information the first time you're getting systematic feeds of sample information from central labs. Unless you don't have a central lab, then you have to deal with sites, and that's fine. You use the manifests. And then central labs may derive samples, so you have new samples created there. They ship, they receive. Sometimes they send you patient information as well. Testing labs, sometimes they derive samples, sometimes they don't. They'll send you information about sample status. They ship, they receive. Storage, same thing. But for future use, you're likely requesting from long-term storage. On the operational side, clearly you need to be able to track lineage of samples. You may or may not need to track storage. If you're managing your own operations, you do. If you're outsourcing everything, you don't. And then various workflows to allow you to request samples, destroy samples, and so on, and follow a standard procedure. And clearly, barcoding, labeling, and attaching data and results. Because reality is very messy compared to the plan, it's important to not only receive data, but also being able to work with data. Because the data you receive is going to be very tricky. You need a platform to be able to turn that sometimes unexpected unexpectedly tricky data into something you can work with. So you need to have a query and reporting engine to be able to do all the cleaning, standardization, and so on before you can work with your data. So one of the things that we've done is to have developed a technology called Chigram, which stands for Query Diagram. And we use this technology very, very extensively, and that basically becomes our querying and data reconciliation and data conditioning engine that is totally configured. Nothing, you don't have to hard code it, or at least significantly minimize hard coding in order to be able to deal with unexpected messiness of what you have coming in from the real world. So some very quick overview of the types of questions that you should be able to answer in that distributed ecosystem in the context of samples and consents. You're largely able to answer this type of questions on subjects because EDC, CTMS, IVR systems are designed to answer this type of questions on subjects. But really, there isn't a whole very good solution out there that's established to answer this type of questions around samples. Okay? So I'll just run through them quickly. You can download the slides and study them offline. So you need to be able to answer the planned sample collections, what am I supposed to get? The actual sample collections, what do I actually have? The reconciliation between planned and actual so that you can act on it if something goes wrong. You should be able to anticipate upcoming sample collections because you know the patient came in for certain visits and certain samples should have been collected. If you don't see it in your data feeds after a certain period of time, something is wrong. Okay? You should be able to see allowable use from the computable version of the consent now that I described. Of what can I do with it? Can I study my sample? Can I use my sample for this indication only or any indication? And so on and so forth. You should be able to review chain of custody. Did the samples go to the right place? And at study closeout, you should be able to look at metrics to study, to look at the study critically from a sample-centric perspective, just like you already do from a subject-centric perspective. And then once you start to build up a library or a database of multiple studies from a sample-centric perspective, then you can start to evaluate, for example, sites based on their ability to deal with samples. When you start a study now, you look at site selection. You probably don't look at their ability to deal with samples very much in site selection. Contrast that with the fact that samples are becoming really, really important, especially for precision medicine trials. Shouldn't you be looking at sample aspects in site selection as well? Right? This is what's going to allow you to do that once you start to have such a library of past performance. And again, exploration reports to drive sample destruction and so on, that should all be automatable with the framework. And this is just an example of the types of, this is not real data obviously, the types of charting that you can do. Amelia is going to go into a lot more details on actionable information about, in this case, expected versus received. That's everything I have. Amelia is coming up next. The organizer, you can switch the presentation rights to her.
Thank you very much, Dan. That was an excellent talk. Can everyone hear me? I hope. Okay. So I appreciate the opportunity to be here today to talk about a topic that has been an ongoing pain point in my career in both academia and industry, and has really been something that I've focused a new company on solving. So it's exciting to be here and to partner with my good friend Jan on this topic. So put this slide up just to say that I worked for a very long time in academia and the pharmaceutical industry, and I was always hired into positions where I was asked to be a scientist, to lead a scientific program. And in point of fact, in order to do my job, I ended up having to build operations from the ground up to support specimen-based research. And also, as the years went on, more years than I like to admit, but thank you, Joanne, for sharing them, um, I ended up having to build informatics infrastructure to support the large amounts of data that we were putting together. And I spent a lot of time doing cross-industry work, looking at how different companies and different institutions approached collection and management of specimens. I worked on an international project with ICHE 16 to really talk about how internationally we're collecting and managing specimens. And it was fascinating work. It's still evolving. But what I was really frustrated with in each of my jobs was that there wasn't really a market solution. I am responsible for having decommissioned some multi-million dollar projects that had been installed over 5 years. Because they didn't work, because they weren't solving the problem that we needed for specimen-based research. And so I love this quote. It was from BioIT in 2010, and I love to highlight that date because the quote was that companies that have access to millions of highly annotated biospecimens with clear consent, traceability, and tools to rapidly mine for desired profiles will have an edge in biomarker-based discovery, segmenting patients for clinical trials, and developing companion diagnostic and theranostic applications. And I think it would be really interesting if the line were open, and maybe during the Q&A session we can get opinions on this, but to know how many people think we've actually achieved this, how many people think their company really has a firm grip on this. And I think it's really interesting that in 2015 we're still struggling to support this goal. Specimen-based research is obviously incredibly important for ongoing research. It enables rapid research to answer questions that emerge during clinical drug development, reduces the cost of conducting prospective clinical trials, but is also an emerging area of complex, focused international regulation, and anyone working in this area is really concerned about the ongoing discussions around the EU data privacy regulation, the possible disruption of safe harbor and transfer of data from Europe to the United States, and even some of the new NPRM provisions for specimen-based research could be real game changers as we're using specimens internationally. Sorry, I'm having an advancement issue here. There we go. The agencies that regulate drug approvals have uniformly advised pharma that at a minimum they should be collecting genetic samples for future testing to really identify genomic biomarkers that identify subpopulations of responders, either those patients with adverse events or those patients who respond differently than the rest of the population. And ICHE 18 right now is really focused on a cross-regional guidance to industry for collection of specimens at baseline in every single patient for future research. As you know, that effort is going to be significantly disrupted possibly by the EU data privacy regulation, which calls for explicit consent for specimen-based research. And it'll be interesting to see how that all evolves. So why is there conflict? What are international ethics committees concerned about? And it used to be that international ethics committees were concerned about use of specimens, but in point of fact, the regulation and law that has been enacted has largely been around use of data, and we're seeing more and more restrictions on transfer or housing of data across borders. And so when we look at their key concerns, the first is, where are the specimens and data going? And is the data privacy regulation in the jurisdiction for which the specimens or data are being transferred to? Are they as stringent as ours? Do they follow the tenets of our beliefs on data privacy regulation? And it's very concerning that recently some of the German data territories have actually called out U.S. HIPAA as not being strong enough to support European data to come to the United States. Now, that's not an official position, and it's not currently disrupting data transfer, but it is concerning and something to watch. There are other countries that certain countries have developed concern about. There are concerns about data going to Southeast Asia in some cases, and all of those concerns are expressed in how data privacy regulation and restrictions for migration of data outside of country borders can be managed. They worry about the ability for individuals to exert autonomy, so to really decide whether to participate in specific research when the breadth of use is large, and especially as we're starting to create whole exome, whole genome data sets. The data sets themselves are very large and contain more data than is typically being analyzed. And so how will that data be managed, secured, stored, are all questions that they're asking. They ask questions about what is the actual risk to our national population if we allow this individual to be analyzed. So really almost counter to the goals of precision medicine, which really want to target the right drug at the right dose to the right patient at the right time. Data privacy regulators are asking, are we putting our entire population at risk by identifying genes or biomarkers that are associated with an increased risk of certain types of diseases like alcoholism, like mental illness, like possibly Alzheimer's disease, and how does that impact our use of data? They worry about national access to research specimens and data. This is a growing concern, especially in third world countries participating in research, but it's even becoming more of a concern in all countries. Across Africa, there's an increased movement for their national researchers to be able to participate in research. Brazil has asked for their researchers to be able to participate in research. And so there's a growing momentum that research should be global, and it should include researchers from that country. There are concerns about coding, the level of coding that's being performed, how data will be shared and who it will be shared with, and ongoing data privacy. There are back and forth debates about whether one-time consent is possible or whether we need to enable dynamic informed consent, whether you need to allow withdrawal of consent or return of research results. And then Brazil, Germany, and the EU continue to evolve their positions on what's important for data transfer. But all of these things really boil down to a key issue, which is trust. Do we trust the group that's coming and using the data to use it in an ethical way? And when we talk about companies that are collecting and using data across borders, they have concern that restrictions are limiting the advancement of medicine that we're trying to achieve with specimen-based research. And so as a third party conducting research, they typically don't have ongoing access to participants, so recontact is difficult unless there's an automated solution. There's a global focus of research, so there are thousands of sites in anywhere from 40 to 120 countries, and it's logistically complex to maintain. New platforms generate more data than analyze, so do we need to analyze portions of the genome, for example, that were not part of our original analysis as a service? Cross-therapy area research is becoming common, and in some cases it's not allowed or heavily regulated in some countries. And all of these things also boil down to trust. How do we as a corporation become a trusted partner for conducting medical research? And so how do we bridge these concerns? So really there need to be new approaches to research, and they need to be pipeline-wide approaches, not one-off protocols, not therapy area-specific approaches, but they really need to be pipeline approaches to build reputation as being an ethical provider of medical research, to manage governance procedures that are imposed by international health authorities and ethics committees, to assure that there are ethical practices for uses of specimens and data, publication, making data transparent once it's complete, and really taking action to make sure these things happen across programs. So what I saw when I was working in the pharmaceutical industry is when we're enabling clinical trials, we perform our operational efforts in spurts. For retrospective analyses, there's a little bit of work to set up the original protocol or collection of the specimen, and then really no work is done until we decide that there's a hypothesis and we're going to generate a data analysis plan and we're going to try to pull the specimens and data. And in some cases we've set ourselves up well to do that, and in some cases we haven't. For prospective analysis, exactly the opposite. We do a sophisticated data analysis plan. We design our trial to collect the specimen for the biomarker that we're planning to analyze. We assure statistical significance. We assure the specimens are collected, and then there's sort of a gap in operations until the end when we have to perform a lot of operations to finalize the analysis. And unfortunately, this sort of sporadic operational approach, which is really because it's often not well staffed and it's not well understood what the operational effort needs to be to manage this process over the course of a clinical trial, is that some data or some operations are ineffective, and the analysis is then either not reaching statistical significance or it's not possible. And so our company was really built to build proactive ongoing operations across the pipeline to assure that the informatics tools, the site-based approaches, the discovery-based approaches are all integrated in a cohesive way to supply the outcome that the company needs, which is really identifying biomarkers that can identify the patient populations that will respond to drugs and respond without adverse event. Understanding that from the time you have an idea that a drug could work all the way back at target identification and taking it through final report to the FDA, there's a multi-step process. There are lots of different types of data and samples that are collected throughout this process. Understanding how to operationalize that is pretty much a missing gap in the marketplace. And then how to take all the data that you generate from these disparate sources and pull them into a foundation that's supportable for translational medicine has been a real gap in the marketplace for some time. So here's a case study we worked on. We were working with a client that had an Alzheimer's development program in early stage development. They were conducting phase 1 trials, and they had a rare skin adverse event. However, because they were using really proactive operations, they had been able, even in Alzheimer's, which is often a difficult program to collect specimens in, they had collected DNA samples in 100% of the patients on the trial. They found a genetic association, they wanted to perform a genetic association study to look for HLA association with the skin rash, and only because they had 100% collection were they powered to detect this rare association, and they made a no-go decision at proof of concept, which they valued in terms of their pipeline recovery cost at roughly $1 billion. So really good management, being able to identify an adverse event, being able to make an early no-go decision really saved a substantial portion of money for that company. There's long-term value for building expert external automation. So you can build that integrated translational medicine database with biomarker and clinical data annotated with consent for every single sample in your inventory. You can assure that you have high specimen collection rates. Jan talked about missing specimens. If you can't see the specimens until the rec form is delivered, you're too late. You're not able to make in-life interventions. So you really need to have live feeds and live intervention capabilities. Your operations change from being sort of a passive recipient and reporting function to being a live, ongoing, interactive operational function within your company. Your management of continuously changing global regulations becomes possible because you're monitoring and annotating specimens as they were collected from where they were collected over time. You have ongoing expert lab interactions to assure your data is complete, on time, and formatted appropriately for use. And you lose reliance on that internal prioritization for software upgrades because, unfortunately, these are not typically the highest priority enterprise systems to support clinical drug development. And so because they fall kind of mid-tier, those are the perfect types of systems to outsource. Data acquisition is complex, and it really does need to be managed by an expert team. Pulling in data, I'm not going to go into this in great detail because Jan did a fantastic job of doing this earlier, but really kind of understanding all the data sources, being able to integrate and pull that data in and integrate it in an ongoing way so that it's actionable, really creating robust in-life data reconciliations that operations teams within companies or within academic institutions can look into and can see that there are missing specimens, that there needs to be an action at the site, and take action. Finding your specimens and derivatives throughout the world is obviously key. It really starts at the site. I know many operational programs are set up to identify specimens once they arrive at the central lab, but that's, again, often, in some cases, 30 to 90 days beyond collection. So that's really too late to make any intervention if the specimen was improperly collected or if there is a problem with a missing specimen. And then you have to track the samples to receiving labs, track them through generation of derivatives, familial relationships with unique codes, and track them throughout the specimen life cycle, which is well beyond the end of the clinical trial. And making sure that you can supply that data to data privacy officers or to researchers around the world who are trying to access the specimens for research. Being able to look across sites during the clinical trial and find out how many specimens are being collected, where they're being collected, and where there are operational issues is very important. And also being able to make interventions. So in this particular case, you're looking at countrywide collection rates, and you see that in this visualization, Peru and Spain have very low collection rates, and there needs to be an intervention in those two countries. There is also the ability to ask the question, because you can geographically and visually see where your specimens are and where they should be. Ask questions like, okay, well, they're only planned to be 100 patients between Spain and Peru, and this is a 10,000 patient trial. Do I care that I have an operational problem in those two countries? Well, maybe I do, because maybe the ethnic representation will be associated with an adverse event, so maybe I do need to make an intervention, or maybe I don't. And so I think being able to make those decisions based on data rather than react to them operationally long-term is important. And then drilling down, understanding, what sites are performing well, what sites aren't performing well, and then tracking site performance over time, knowing how those sites perform in different therapeutic areas at different points of time is really key to performing well. This is another case study where we worked with a client in late-stage development. When they were managing an HCV filing, they actually had fantastic sample management, really high collection rates, and what happened was mid-phase 3 the scientific landscape changed. A new biomarker was identified that was associated with response, and they were in a race neck and neck with another client for filing first for this new mechanism of action for a drug. And in this particular case, the company had fantastic strategy. They had collected samples across the trial. They had high collection rates. So when the landscape changed, they could operationalize and perform an in-life analysis using future biomedical research samples. The other company could not. And so this company got to market first, and their marketing team estimated that this was roughly $1 billion advantage to not only the product itself, but all of the operational costs that would have been involved with sending out informed consent and getting new consent to collect specimens, getting those specimens in, and then analyzing them. if you really look at data banks and biobanks that have high return on investment, they're data banks that represent broadly consented samples, largely representative of clinical trial populations over time. Most clinical trials are included, so it's not a subset of trials or only phase 3 trials or only oncology trials. It's all trials, and you can pool across protocols and programs. You have uniformly annotated specimens and data sets, and the biomarker data is combined in a central repository with clinical data for in silico analysis. And advanced search and analysis functions are enabled. That's really where you start to build high return on investment. So with that, thank you very much, and we look forward to the questions.
Well, thank you to both of you. That's a really terrific presentations. I'd like to ask you to drill down a little more about the that introduction, that when the sample is collected. We ran a program last summer. One of our speakers was John Wilbanks from Sage Bionetworks, which had been working on a phone-based, very complex, but at least easy to it was not a paper-based consent system. Can you help us have a picture of what your consent system is like?
So are you talking to me, Joanne, or to Jan,
Sure.
or to the audience?
Both of you. So I'm gonna present the questions to you two, and then we can think about asking the questions to the audience as well.
Very good.
Why don't you go first, Amelia?
Sure. So we work with clients who employ a number of different solutions. So some clients use e-consent. Some clients still use paper-based consent forms, but regardless of the format of the consent, how they're implemented at the site and how they're managed at the site in terms of format is less important long-term to the use of specimens as is the conversion of that format to actionable consent. And so, also the ability to construct a globally viable document is very complex and takes significant expertise to do. So it's, we work with clients that use both, and I can't say that either is more effective. I think the programs that are most effective are the programs that have really expert operational support. The format of the consent has not seemed to matter so far.
So so either way, there's a layer of taking that data and then getting it into the barcode and so on. Is that a manual process in some cases? I would guess.
It can be. It can also be automated. I mean, we certainly have an automated solution, but it can be done manually in a very effective way as well.
Great. Jan, did you want to chat about that?
Sure. So, yeah. So there are basically two issues behind your question. The first issue, to me, is the biggest issue, and I agree with Amelia's statement. The biggest problem now with consent is that it's not in a computable format. You have to actually, with your two eyes, read the consent before you can determine what you can do or not do with the samples. Mm-hmm. That's the biggest problem. So we need to extract the information in the consent paper to a form that's computable. So I talked a little bit about it. Now, the second aspect is how do you deliver the consent, right? So currently, I understand it's mostly paper. There are e-consent companies that are starting to get traction. That's a separate issue. So when you design, let's say if the world suddenly switches all to e-consent, that still has the question of, are you providing computable information from e-consent, or is it just electronic paper, right? Mm-hmm. So both aspects are important.
Yeah, I agree 100% with Jan on that, and I think there have been sort of mistaken understanding that a QR code replaces actionable consent parameters, and that's not true. It's a QR code, in my opinion, in most cases is equivalent to a version number on a paper-based consent and really doesn't supply, in most cases, in most installations that I've seen, much beyond that.
Thank you. So I'm intrigued by the idea of someone withdrawing consent. Have you seen a situation, I mean, as a where individuals have actually done that, and has it ever been possible to trace through to all the samples that might be distributed in many different labs? Is that a new concept that really hasn't been done yet? Yeah.
No, it's absolutely happened. It happens infrequently, thankfully, for medical research, but it does happen. And in some cases, it's happened because of news. Henrietta Lacks, the Havasupai case comes out, and you'll have kind of a rash of withdrawal of consent. It absolutely, in a good system, is possible to link back to all the specimens and derivatives and destroy them. If there is a good system in place at the site, I think it's mandatory. You have to have a really good system linked. What's going to be more interesting is that under the EU data privacy regulation, they would also have the ability to delete data, which is in conflict with what a regulated industry like the pharmaceutical industry is allowed to do. So that is an interesting regulatory conflict that we'll see resolved in the future months, I'm sure.
I could see that. Lovely. So we have a way we could actually ask a question. Now here we have a comment. I'll do that, but I'm going to ask a question in a little bit, and I'll ask everyone has on their dashboard they can raise their hands. So I'm going to ask a question, and then we'll ask you to raise your hands to answer it. But first, here's a comment. I have no immediate questions, but want to comment that both presentations were superb. That's very nice. Really hit the nail on the head on all the challenges and issues. We run a central lab, and these presentations will be invaluable in future planning. Many thanks. That's very nice. Would you like to talk a bit about the specific issues, Jan and Amelia, about central labs and the opportunity that such labs have to do things right?
Yeah, I can start. So we work with exchanging data with central labs. It's challenging. The slide that I had with plan versus reality, you literally run into all the issues you can possibly imagine and more that you can't imagine when real data come in. Okay, I'll give you some examples. Basic concepts like visits. Right? How you describe a visit is different from what's in EDC to what's in central lab to what's in testing labs. Things like that, right? That you have to reconcile after the fact. It would be great if the industry can get together to have some standards to reference concepts, critical concepts like visits in a standardized way. Now, I'm of the opinion that standards are great, but if you wait for standards before you move on with life, it's going to be an awfully long long wait. So while I want to try to strive for standardization, I understand that we just have to work with the world as is, and that's what we had to do. The other observation that I had with central labs and testing labs as well is I think they take a very study-centric view of everything. Okay, everything is study by study. Now, the way we're looking at things, you need to aggregate up to a much more global view, and it would be great to have standardization again across studies, at least for a subset of information that's critical for the sample-centric perspective, as I described earlier. Amelia, anything you want to add?
Yeah, I think, we also work heavily with central labs, and I think that from my perspective, it really behooves the central labs, quite frankly, to work with an expert partner, because I think that the regulated landscape and the multiple types of clients that central labs work with really kind of build a fairly complex model for them to interact in. I mean, every client has a different format they want to see their data in. Every client needs something different, and they need their regulated database to be auditable. I mean, it's a very complex world that the Central Lab works in. And so I think for these types of kind of next generation pieces, working with an expert partner is a great solution for central laboratories so that they can focus on their core function, which is, you know, providing expert analytics, and allow the data piece to be managed by an expert partner. Because it's very complex, it's heavily regulated, and it's not really part of it's sort of it's almost like translational medicine. It doesn't exactly fit in discovery, it doesn't exactly fit in clinical, it fits across the spectrum.
Well, beautiful, and we're going to sort of come to a close in a moment, but I do want to ask a question of our audience. And you have a little, there's a little ping you have in your dashboard as if you were just raising your hand, so I'm going to use that to poll. And the question I have is who in the audience feels like your organization, whether it's a small lab, a central lab, or anything in between, who feels like you're doing a very good job at this tracking, consent tracking, essentially?
And sample tracking.
Okay. So we could say sample or consent, because both of those are very difficult, but I don't see any hands raising. So I think this is an interesting topic that I'd like to come back to. I am going to ask the two of you one more question, though, is what would be the entity that would convene the different parties to develop a harmonized approach while everybody's doing their own thing, but developing a understanding amongst everyone? Is there an existing organization, a NIST type approach, or is this
for data standards? For data standards related to clinical trials, I mean, CDISC is what most people refer to, but the biomarker piece and some of the clinical data pieces are not standardized, and they're on their planning pipeline, but they're not standardized. So there have been some efforts to move ahead and develop some standards that allow integration of data at different, you know, stages of the pipeline that are not covered by CDISC, and those efforts are being published, I believe, soon. There's an effort, we worked on an effort, I know Duke University has worked on an effort, Harvard has worked on an effort, so there are some different groups that have really contributed a lot to harmonizing data standards.
Terrific. Well, thank you so much. I would like to just give each of you an opportunity to say any final comments you'd like to share. You want to start first, Amelia?
Well, I just want to thank you for this opportunity. This is obviously a topic that is near and dear to my heart, and I think there's a lot of in this space, thank goodness, and there's a lot of new tools and approaches to solving these issues, and so I'm excited about the future, and even though it's a complex environment, I think that there's a lot of novel approaches that are already being applied that can be great solutions.
Thank you. And John?
Yeah, I would echo what Amelia just said. It's a very exciting field. I think precision medicine really made the advent of precision medicine is making this problem we're talking about today a must-have instead of an optional problem to solve, right? You know, mishandling a few samples in a gigantic trial is one thing that may or may not cause any harm, but mishandling a few samples in a much more targeted trial where, you know, several tens of patients may determine, you know, a billion-dollar drug or not, that is a very different problem. So I wanted to raise awareness within your organization that this is an issue that you need to address. And, you know, today we only talked at a fairly high level, and more than happy to chat more in more details if you'd like. Thank you all.
Well, thank you to both of you. Thank you to our audience. Thank you to our tech crew. And have a wonderful holiday season, everyone. And we'll be back in action with you again in a few weeks. Take care.